metabolic hormone, which we're going to see is going to be really important today for fat synthesis. Some people are leaning more towards carbs versus fats for energy, perhaps, more muscle versus liver, but your liver definitely is super important. And we're going to see a little bit more about that next week when we start to talk a bit about gluconeogenesis, which I'm going to mention a couple times during today's lecture. Really, I'm trying to get you ready for the final exam, okay? I'm going to do my best to review as much as possible. It's coming up really quickly, so hopefully over the next 2 weeks, you're staying on top of things. Keeping up with your review, we'll do another ATP calculation today as well, and you're going to see just how much energy is going to get made from our fats. Okay, so a huge reserve of ATP, and we'll do another calculation just to help get you thinking through that, because I guarantee you there's going to be an ATP calculation on the final exam. So really, again, we're going to learn some new pathways, just like with the carbohydrate metabolism that Dr. DeMello talked a bit about. It's about understanding the purpose of the pathways, understanding how they're regulated, and this is going to involve those hormones. So we're going to talk a bit more about the signaling pathways again. Okay, really that reciprocal regulation of turning one thing on and the other thing off so that they're not both active at the same time. So this version of the simplified map is going to be on the final exam. So you will have it when you sit down in the final exam. You can sit down, you can annotate it for your own purpose. You can't bring a version into the exam. It's not a cheat sheet to bring with you, but it is something for you to do a knowledge dump when you get in. Okay, so I would suggest you do that. Write down everything you remember about all the pathways, all their regulation, so that when you get to the multiple choice questions, you're not confused about some of the statements. Okay, and so the final exam will be multiple choice as well as short answer. And you can use your review questions from tutorial. Hopefully you're doing those ahead of time to help practice how to answer some of the short answers in our course. Yes, quick question. Louder or not louder? Louder. Okay, we want louder. All right, are we okay in the back there? All right, thank you. All right, so let's review a little bit about our fat molecules. So this is coming back to the start of the second half. Remember, our final exam starts with our membranes and our lipids and our membrane proteins. One of the classes of lipids that we talked a bit about were the triacylglyceride molecules. These have that pink glycerol backbone that's just shown on this slide, and this is how we store carbons for later. Okay, so if we don't need to generate all that energy from maybe some of the carbohydrates that we're consuming or even any of the fats that are in your diet, we're going to store them in this molecule. Okay, so we have these 3 acyl chains that are shown in blue, so we call them acyl chains or fatty acid chains. These are usually in the reduced form, so they're saturated chains, so no double bonds. We learned about double bonds and the importance of that for fat structure, maybe in your membranes, but when it comes to storing fats, the triacylglyceride molecules are saturated with regards to these 3 fatty acid chains that are attached. So we have that ester bond and linkage to our glycerol backbone, and this is what's going to allow your adipose tissue to accumulate these carbons or store these carbons until we actually need them for energy. The thing, though, is that we first need to hydrolyze them off, so we need to release them from this triacylglyceride molecule. You might also hear the word triglycerides or TG for short. These are just different ways of describing the exact same molecule. So when we need to use these fats for energy, if you're thinking about, say, an adipose tissue or fat tissue, which has this unlimited store of triacylglyceride molecules, we really do need to have some hormone signaling occurring first in order to release those fatty acid chains to be broken down for energy. So those hormones that we talked a bit about, epinephrine or adrenaline, that flight or fight response hormone that's going to help generate energy, is going to signal by our GPCRs, so those beta-adrenergic receptors, or glucagon, which is our low glucose hormone, is going to signal that we don't have glucose for energy, so instead we're going to start breaking down our fats for energy. And what that's going to do is it's going to initiate that signaling pathway that we talked a bit about already, where we've got adenyl cyclase that's going to get activated by that G-alpha subunit of our GPCRs, producing our secondary messenger, cyclic AMP, that will then activate protein kinase A, and then protein kinase A will phosphorylate our lipases, and these are then going to be activated to help break down your triacylglyceride molecules. Okay, so we have hormone-sensitive lipases, we have lipases called our triacylglycerol lipases, all have that conformational change that's needed so that the enzyme can now use a water molecule to hydrolyze or break off those fatty acid chains. So you're going to start with your triacylglycerol molecule. We're going to use a water molecule to remove one of the fatty acid chains. When you remove one of those, that creates our diacylglycerol, so 2 acyl chains attached to glycerol. Another water molecule comes along, and your lipase can cleave off another fatty acid chain to release monoacylglycerol, and then there's going to be other lipases that can eventually release that third one to produce glycerol, which we'll see next week can get used to make glucose. And your free fatty acids can then get transported to other cells that can maybe use them, say your muscle cells. Okay? But if you remember that structure of our fatty acids, so up here, just my little cartoon version, remember we had our carboxylic acid group, that little red polar molecule, then we had these long hydrophobic fatty acid chains, the tail portion, creating that amphipathic molecule. We do need another molecule called albumin, which you might have heard about, another protein that's going to help transport these amphipathic molecules throughout the bloodstream. Right? So we have albumin that's going to help take it to where it needs to be used. So sometimes people might go in for blood work and you might have albumin ticked off as a level that might get tested, because it is a very important protein that's going to help transport not only these fatty acids, but also things like other vitamins and other hormones that might be needed throughout the body. So we've now mobilized or released those fatty acid tails, and now they can get used for energy. Okay, so one thing, when it comes to fat metabolism, this mobilization does take a little bit of time, right? Earlier, when we were talking about carbohydrate metabolism, your muscle cells already had that glycogen in them to use that the glucose for energy. But when it comes to fats, you do need to mobilize those fatty acid chains so that other cells can use them. So just to review, in words, we've got our catabolic hormones, epinephrine and glucagon, that are going to bind to our GPCRs on adipose cells. That's going to initiate our cascade to release our G-alpha subunit to activate adenylyl cyclase. That'll produce cyclic AMP to activate protein kinase A. And then that kinase can come along and transfer a phosphate from ATP onto different hormones like triacylglycerol lipase or hormone-sensitive lipase, and that's what's going to activate them so that they can now carry out that breakdown of our triacylglyceride molecules. We've got our tags that have 3 fatty acids on them. We're going to use a water molecule to hydrolyze off those fatty acid chains, and now they can be transported via albumin to the other cells that are going to use them for energy. So this is just showing you that in pictures. So on the left, here's our fat cell, our huge storage form of carbons. We're going to break down our triacylglyceride molecule to glycerol, which can be used to make glucose in your liver cells. We'll see that next week. And then our fatty acids can maybe either go to the liver cells for energy or muscle cells that might have a huge energy requirement, say when you're exercising. And we're going to see how those are going to get broken down in a process called fatty acid oxidation or beta oxidation in order to produce acetyl-CoA, as well as our reduced coenzymes NADH and FADH2. And that's going to produce a huge amount of ATP in oxidative phosphorylation, as Dr. DeMello covered last week. All right, so let's just do a quick question. So there's different types of molecules that can be found in the body. There's acetyl-CoA, which we've seen already, and then there's also acyl-CoA. So take a minute with your seatmate and see if you can recall the difference between acyl-CoA and acetyl-CoA. All right, we've got a couple people that are picking the first 2 options. Acyl-CoA can be used for energy, but acetyl-CoA cannot. Acyl-CoA has 1 less carbon than acetyl-CoA, but the majority of people are going for acetyl-CoA has 2 carbons attached to CoA, while acyl-CoA is a longer chain, and that's going to be our correct answer. Okay, so make sure you remember the difference between these 2 words. They are quite similar, but this is one of the typical errors that we notice in, say, the short answer section of the exam, right? If you're writing a sentence and it says acyl-CoA instead of acetyl-CoA, they are very different molecules, and it can make your answer correct or incorrect if you pick the right one or the wrong one, okay? So when it comes to these 2 things, acetyl-CoA has that structure of our CH3, C double bond O, to a CoA, and then your acyl CoA is going to have a longer fatty acid chain that has multiple carbons in it. Okay, there should still be that C double bond O. Okay, and so the first thing that needs to happen is when our fatty acid chains get to a cell, we need to actually activate it and add that CoA molecule on here. Okay, so your acyl chains are being transported through the bloodstream via albumin. We've got these amphipathic molecules that are getting to the cell, and we need to have a way of kind of trapping them in the cell so that they're not maybe stuck in the membrane or binding to maybe some other hydrophobic molecules. And so the first thing that happens when they get to the cell is they're going to get added onto that CoA molecule. So it's going to be a covalent addition, so we're creating a new bond between that acyl chain, so just represented down here in green, where R is a longer chain, so R is going to be our long chain of carbons. And we have an enzyme called acyl-CoA synthetase. Again, tase tells you that it's an enzyme. This one's actually going to use energy in order to create that covalent bond. So we have our coenzyme A that's going to get added on to where our free carboxylic acid group is, and that's going to use an ATP molecule. And here's an example, another example of where ATP is going to get hydrolyzed to AMP. So it's not getting hydrolyzed to ADP, it's getting hydrolyzed to AMP in a reversible reaction. So here's our reaction mechanism with our enzyme acyl-CoA synthetase. It's fairly reversible in a cell to create that new thioester bond between coenzyme A and our fatty acid chain. But it is reversible, so the question I have for you is, how can this be favorable in a cell? How is this reaction going to be favorable in a cell based on what we're seeing? The use of ATP in order to add that coenzyme A on it. What's going on with our products here? That's another meeting I'm supposed to be at right now. But I'd rather be here with you. There we go. Yeah. Yep, so what happens to the phosphates? All right, yeah, good. We're going to drive our reaction in the forward direction. So if you remember from tutorial last week, talking a bit about this pyrophosphate molecule, we can actually hydrolyze it again. So we're going to take another water molecule. That's going to come along. Lots of water in the cytosol, right? These products that are being made are in solution, surrounded by water. That water is going to come along. It's going to hydrolyze it into 2 phosphates, and that's what's going to help drive our reaction in the forward direction. Okay, so that's going to drive this reaction, so it's quite favorable. And you might ask, well, why does this happen? You know, it just depends on the enzymes, right? It depends on their mechanism, which you're looking at a little bit in your assignment as well, in terms of what products are actually going to be made. Okay, so we're going to take our acyl chain, we're going to add on that coenzyme A molecule, we're going to use ATP as our energy molecule, producing AMP as well as pyrophosphate, which then gets hydrolyzed to 2 inorganic phosphates, driving it more into the forward direction. All right, but there's that problem, which you might have talked a little bit about in tutorial. We've kind of seen a couple times coming up throughout the weeks. When we actually produce an AMP molecule, this is an energetically inefficient step. Okay, so the majority of ATP that gets remade is made by ATP synthase, which you looked at with Dr. DeMello. ATP synthase can only use ADP as a substrate. It can't use AMP. So what we need to do is we need to first convert that AMP back to ADP. And what we're going to do is we're actually going to sacrifice a second ATP molecule. So we have AMP that was made by acyl-CoA synthetase. We're going to take our second ATP. So here's our second ATP. Okay, so the second ATP is going to come along. So the first AMP is coming from our activation. Our second ATP is going to come along, and it's going to donate one of its phosphates onto AMP. So adenylyl kinase transfers phosphates from one molecule to another. It's going to be transferring it amongst our adenylate nucleotides, transfer the phosphate onto AMP, and now we have 2 ADP molecules that ATP synthase can use in order to regenerate our 2 ATPs. So this means that if you're trying to figure out how many ATPs are made or required in any of these pathways, when it comes to fat metabolism, whenever you are activating your fatty acid chain with a coenzyme A molecule, you're actually going to require 2 ATPs. So one for that activation step, and then one to regenerate ADP from that AMP molecule. Okay? And then as we saw in the previous slide, you're also going to require 1 water molecule to hydrolyze PPI. Okay, so just keep these in mind when you're going through your calculations. Yes? So the question is, when it comes to these irreversible versus reversible reactions, right, when it comes to activation, this activation step, we said that it's usually reversible if you just consider the reaction on its own, okay? But you have to think about these reactions within the cell where there's lots of other things occurring, okay? Just going back to our thermodynamics portion of the lectures, remember it depends on what the concentration of substrates and products are, right? For your delta G, those delta G reactions are dependent on how much substrate and how much products there are. If you're going to pull away your product, if that PPI is going to get hydrolyzed and pulled out of that reaction, then you actually have less product, and that's what's going to drive your reaction in the forward direction. Okay, so if you didn't have that happening, then it might be reversible, and your acyl-CoA might want to go in the reverse direction. But as soon as you start to, you might be going in the reverse direction, but as soon as you start to deplete some of those products, that's what's going to help promote it in the forward direction. Okay, so thermodynamics is really going to help dictate a lot of these reactions. All right, so we have our acyl-CoAs that are being made. This is all being made in the cytosol, okay, or in the cytoplasm. And when it comes to the actual breakdown and energy production, that's all going to happen in the matrix. Okay, so as Dr. DeMello talked a bit about mitochondria last week, remember we have these porins, these pink membrane proteins that allow molecules to cross that outer membrane, but then we have this blue inner membrane that's quite impermeable to different molecules, right? That's where we were setting up that proton gradient for ATP synthesis to occur. So we do actually have to have transporters that are going to move those molecules into the matrix in order for breakdown to occur and energy production. So those acyl-CoAs come in from the cytoplasm, beta oxidation or our catabolic pathway is going to occur inside of the matrix, and that CoA molecule is quite large. It can't pass through the inner membrane, it can get through the outer membrane based on porins, but we do actually have a transport system that's going to help move it into the mitochondrial matrix. Okay, and so you might wonder, well, what's the point of actually making these acyl-CoA chains outside of the matrix if it's only used inside of the matrix? Okay, and the answer is really regulation. There might be some rules for it in the cytosol, but if we can dictate whether or not they're coming in or out of the mitochondria, then that's going to regulate or determine whether or not fats are going to get broken down or be used for other parts of the cell. Okay, so your catabolic pathways are all going to take place for fats. Beta oxidation is going to take place in the mitochondrial matrix, and as long as you get the fats in, then they're going to get broken down for energy to be made in beta oxidation. So this is where something called the carnitine shuttle is going to come into play. There's going to be 3 components or 3 proteins that are a part of this shuttle. Okay? So if we start on the cytoplasmic side, or sort of the intermembrane space up here, okay, so you can think of cytoplasmic side as the same as the intermembrane space. We had our acyl chains being added on to CoA to produce acyl-CoA. And that CoA molecule is too large to get across the membrane. So what we're going to do is we're going to move it onto a molecule called carnitine. Okay, so carnitine is a much smaller molecule. It's easier to get across that inner mitochondrial membrane. And we're going to have a transferase. So this is why we learned about all the different classes of enzymes. Right? So we have a transferase that's going to transfer that acyl chain from CoA onto carnitine, releasing a free CoA molecule as a product. Okay, so the first component of our shuttle, the first protein that's going to play a role is called carnitine acyl transferase 1, or CAT1 for short, and that's going to create our acyl carnitine group. We then have a translocase, so here's an example of a membrane protein in the inner mitochondrial membrane, and this is going to be an antiporter, so it's going to move one thing into the matrix in exchange for another thing. So it's going to move our acyl carnitine molecule in exchange for a free carnitine out. And when that free carnitine goes out, then it can get used and joined onto another acyl group for import. Now that we've got our acyl carnitine inside in the matrix, we're now going to transfer it back onto our CoA molecule. So there's a second transferase, CAT2, that's now going to move the acyl chain back onto a free CoA in order to reform our acyl-CoA. And now we've got our acyl-CoA chain, and that can now get used and catabolized in beta oxidation. All right, so I have a question for you. Do you think carnitine supplements, so if you took extra carnitine, do you think this could act as a magic pill to help promote weight loss? So take a minute with maybe your partner and think about whether this could promote weight loss if you were taking extra carnitine. All right, we've got a lot of people saying no. Is anybody brave enough to argue for that? I have the very scary microphone. This is your chance for a little bit of fame in BCH 210. Is anybody brave enough to argue yes or no? Or maybe? Thoughts?
It's not actually going to give you any more acyl-CoAs to transport through.
Okay.
So you're not really getting any more energy.
All right, so it's not going to give you any more acyl-CoAs. Where are those acyl-CoAs coming from?
They're coming from your fats.
From your fat cells, right?
Yeah.
And how do we get them out of the fat cells?
You get them out of the fat cells using epinephrine or glucagon signaling.
Epinephrine, good. So using this thought process backwards, right? You could take these carnitine supplements, and they could maybe move those acyl chains into the mitochondria to generate energy, but if they don't have that starting substrate to begin with, you're really not going to be breaking down fats. Okay, so no, or maybe you could say maybe they might work, right? As long as you're exercising, you've got that epinephrine signaling that's going to start breaking down your triacylglyceride molecules, then that might provide some of the substrates to help with breakdown. But if you don't have that, then it's really not going to act as like sort of that magic pill to promote weight loss. Okay, so thank you for your answer. All right. So we'll say no, or sort of maybe. You really need acyl chains first, acyl chains for beta ox, right? And those come from epinephrine signaling. All right, so once we do get the acyl chains in, we're going to see a series of steps in beta oxidation, okay, where we're going to start breaking down that long fatty acid chain. Okay? And there are 4 steps in beta oxidation. You don't need to know the enzymatic steps. Again, it's all about the overall purpose of the pathway. Okay? So taking our long acyl-CoA molecule, so here at the top, remember the convention for how you should remember and be able to recognize our fatty acid chains. We have 2 different numbers. The first number tells you how many carbons there are, so 16 carbons. And the colon, and then the 0 tells us that there's no double bonds. Okay, so with regards to this, the purpose of this course, we're just going to think about chains that are saturated in terms of being able to break them down for energy. All right, so we're going to start with our fatty acid chain, and you can count all the carbons and see that there's 16 carbons in there. And when it gets into the matrix, there's going to be a series of 4 reactions. That's just what this little icon is showing you, 4 little arrows that are going to take that chain, and we're going to remove an acetyl-CoA molecule. Okay, so those 4 reactions are going to require a water molecule as well as another coenzyme A. Okay, so here's a little blue CoA coming in. It's going to join onto our chain, and then we're going to release the pink CoA that was on the longer chain as acetyl-CoA. Okay, so we're going to release a free acetyl-CoA each round, as well as making NADH and FADH2. Okay, so each round of beta oxidation is going to break down our chain, and we're going to release acetyl-CoA, which has 2 carbons, and now our chain itself is 2 carbons shorter. So we started with a 16 CoA molecule. Here's our product, 14 CoA. Along with the production of NADH and FADH2, we can generate lots of energy from those molecules in the electron transport chain and oxidative phosphorylation. So those are going to go to ETC. Where does the acetyl-CoA go? What pathway does it go into to generate energy? TCA, I think I heard. Yep, good. Some people are reviewing that. That's great. So that's the first round of beta oxidation. Now those 4 reactions are going to repeat. Now we're going to start with our 14,0-CoA molecule. Again, water molecule comes in, a new CoA molecule comes in that's going to release an acetyl-CoA as well as producing 2 NADH two reduced coenzymes, so an NADH and FADH2, to produce our 12-0-CoA. So our chain gets shorter by 2 carbons. Third cycle, so that's first time, first cycle, second cycle, third time. Again, water comes in, CoA comes in. We get 4 reactions to release acetyl-CoA as well as an NADH and an FADH2. And the cycle is just going to keep repeating until the very last cycle. When you start with a 4-carbon chain, you're actually going to produce 2 acetyl-CoA molecules. Okay. So if you start with 16-0, how many rounds are we going to need? So how many rounds of beta oxidation are we going to need in order to break down our 16-0 chain? All right, we got majority of the classes going with 7. Okay, so 7 is going to be our correct answer. So let's just take a look at that. I can switch my slides back. All right, so we talked through the first couple of rounds, right? Round 1 makes an acetyl-CoA. Round 2, round 3, where we ended with a 10-0-CoA molecule. Okay, so let's start with that 10-0-CoA. So round 4, we're going to break down our 10-0 to make another acetyl-CoA, as well as an 8-0-CoA chain. Round 5 is going to break down your 8-0 to 6-0. Okay. Round 6 is going to produce our 4-carbon chain. Okay. And then when you break down your 4-carbon chain with CoA on it, right, you only have 4 carbons left, so we're going to make 1 acetyl-CoA, and then your 2-CoA is essentially acetyl-CoA. So I guess you could call this 2-0-CoA, right, which is really the same as acetyl-CoA. Okay, so last round, you're going to make 2 acetyl-CoAs. So if you're kind of like thinking about a shortcut, you can take N equals the number of carbons, and we're going to go N divided by 2 minus 1 for the number of rounds. Okay, just as a little shortcut. All right, so that's going to break down that's how we are breaking down our fatty acid chains. Okay, so last round makes 2 acetyl-CoA. There are 4 steps in beta oxidation, and again, you don't need to know the information on this slide, but I did want to present it because maybe if you're going to med school and you're writing the MCATs and you need to know this kind of stuff, for some people it helps to think through the chemical logic, okay? But the whole point of having these 4 steps is to generate a chain that can be broken, okay? We're starting with a saturated chain of carbons, and they are very stable. It's really hard to break a reduced form of that chain, and so that's what all of these steps are going to do. Okay, so we've got some redox reactions, so some oxidoreductase reactions where our coenzymes are going to get made. So we're going to make an FADH2 that's going to create a double bond. The water molecule, which you do need to remember, so you need to know that each round requires a water molecule. So that water molecule is going to come in. It's going to get added across a double bond at the beta carbon. This is why sometimes we call it beta oxidation or fatty acid oxidation. Then we're going to see another redox reaction to make our second reduced coenzyme, NADH. And then that's going to create a keto group. And then our last step is where that new CoA is going to come in, and we're going to break off our acetyl-CoA to produce our chain with 2 fewer carbons. Okay, so again, you don't need to know the details of all of the steps, but you do definitely need to know the net reaction in terms of what molecules are required. So we're requiring a water molecule and a CoA molecule, and then we're producing an FADH2 and an NADH. All right, so if we started with our 16-CoA molecule in the mitochondria, after we've imported it through the carnitine shuttle, we are going to have 7 rounds of beta oxidation. So you're going to need 7 NAD pluses and 7 FADs, as well as 7 waters and 7 coenzyme A's. And that's going to produce 8 acetyl-CoAs. Remember, we have 2 made in the very last round. And then our 7 NADHs, 1 per round, and 7 FADH2s, 1 per round as well. All right, so this is all going to be important because this is going to help us do our ATP calculations. Okay, so we're going to use this information in this last line. and I'm going to give you some time to walk through the calculation. Okay, so you're going to use this table, and we're going to walk through, and I really would encourage you to do this. Okay, I'm going to walk around, and I'm going to yell at you if you're not working on it in the next 2-3 minutes. Okay, take this table and start thinking your way through this problem. You have like 3 weeks until the final exam. It's better to learn this now than have to cram everything getting closer to the exam. Okay, so I'll get you started. So from this slide, we've got 8 acetyl-CoAs, 7 NADHs, and 7 FADH2s. Okay, so we're going to fill that out first round. First row. Beta oxidation is going to make 8 acetyl-CoAs, 7 NADHs, and 7 FADH2s. And then we are going to require 7 waters. So we'll do a -7 there. Okay, so take some time, put those 8 acetyl-CoAs into the citric acid cycle, and see what you come up with, and use Mentimeter to show me your answer. Okay, so take 2 or 3 minutes to do that. Okay, I see lots of people reviewing your citric acid cycle, right? So that's good. So remember, go back, review the summary. Each round is going to make 3 NADHs, 1 FADH2, and 1 GTP, right? So you're going to have to multiply those numbers based on how many times the cycle's running. So we're going to have really big numbers. You've got NADHs made in beta oxidation, lots of NADHs made in the citric acid cycle. So add those up. Got a couple answers coming in Mentimeter. I'll give you 1 more minute. See if you can keep working towards a final answer. All right, we've got a bunch of people saying 108. Let's see if that's the right answer. Okay, so we said we were starting with our 8 acetyl-CoAs that are going into the citric acid cycle. We've got some NADHs and FADH2s made in beta oxidation. We're going to make some more in the citric acid cycle. I just wanted to bring back this slide from Dr. DeMello's lectures. Remember, oxidative phosphorylation is going to take those NADHs. So we've got the electrons from NADH that are going to go through complex 1 to pump 4 protons, complex 3 pumps 4, and complex 4 pumps 2 to give us a total of 10 protons. 10 divided by 4 gives you 2.5 ATPs for every NADH. Okay? And then our PO ratio for FADH2 is going to be 1.5, so we're going to need those values as well, right? So you're going to walk through the pathways, find out how many NADHs and FADH2s are, and then stick them in oxidative phosphorylation in order to make our ATPs. All right, so we're starting with 8 acetyl-CoAs from beta oxidation, which we saw from our 7 rounds of beta oxidation. Those are going to go into the citric acid cycle. And every time the citric acid cycle runs, you're going to make a GTP, 3 NADHs, and an FADH2. So we're going to multiply all those numbers by 8 to give us 8 GTPs, so 1 per round, 24 NADHs, so 3 times 8 gives you 24. Yes, you'll need a calculator for the final exam if you can't do this in your head. You probably can't do the PO ratios in your head. I have a hard time with them too, so definitely don't forget your calculator. FADH2, 1 per round, so citric acid cycle runs 8 times, so we're going to get a total of 8 FADH2s. And then remember, every time the cycle runs, you're going to require 2 water molecules. So 2 times 8 is minus 16. Everybody with me so far? I see some nodding heads. Okay, good. All right, now we're going to put our NADHs and our FADH2s, those are going to go into the electron transport chain. Okay, so we're going to add those up. We're going to add up all of the NADHs from beta oxidation. We're going to add them to the ones that came from the citric acid cycle. So that's going to give you a total of 31 NADHs that are going to go into the electron transport chain, and then 15 FADH2s. Remember, let's just go back. Sorry. At the end of the electron transport chain, we make a water molecule. So for every pair of electrons that goes through the electron transport chain, we make a water molecule from either NADH or FADH2. So if we add up those two numbers, so 31 NADHs plus 15 FADH2s, that's going to give you 46 water molecules at complex 4. Okay, so we're on our way to figuring out how much water is being made as well as how much ATP is going to get made. Those electrons flow through the electron transport chain. We set up that proton gradient, and ATP synthase is going to use that proton gradient. And so this is where our PO ratios come into play, right? 2.5 for all of those NADHs that are present. So 31 times 2.5 gives you 87.5. 15 FADH2s only give you 1.5 ATPs. So that gives you 22.5. Remember, we don't round down until the very end, so this is just an approximation at this step. But if we add those two numbers together, this tells us that ATP synthase has made a total of 100 ATPs. And every time an ATP is being made, you also get a water molecule being made, right? ATP synthase is using this huge pool of protons to make all these ATPs. You can't really keep track of where they're coming from, whether it's an NADH or an FADH2. They're all getting used together to make 100 ATPs and 100 water molecules. So we're going to add that all together. Remember, any GTPs that are being made in the cycle are equivalent to ATPs. We can exchange those phosphates and not lose any energy. So you're going to make a total of 108 ATP molecules. If we add up our water column, so we have 100 made by ATP synthase plus 46 made in the electron transport chain gives you 146. Subtract off 16 gives you 130. 130 minus 7 gives you a total of 123. Okay, so if you picked 108, then that was correct. The grand total for the complete oxidation of a 16-CoA chain in the matrix is going to give you 108 ATPs and 123 water molecules. Okay. This is starting with your chain in the mitochondrial matrix. If you start with it in the cytosol, remember we were talking about that activation step, we're going to require 2 ATPs and 1 water molecule. So you have to subtract those off. Okay, so the total will be 106 and 122 if you're starting without the CoA on it. So just your free fatty acid chain that maybe came from your fat cells. Remember, we first need to activate it and then get it into the mitochondrial matrix. All right, I'm going to do one last question before we take a break. And I'll put up the table at the break as well. So in terms of fat catabolism, whoa, what's going on here? Okay, so 108 was our correct answer. There are some patients that have a CPT2 deficiency. Okay. And which of the following do you think would occur if someone had a CPT2 deficiency? Okay. Would we see hyperglycemia, hypoxia, insulin sensitivity, or muscle weakness? We've got the answers popping up already. Right, so when it comes to, oh, did I forget to put this in our slides? Is that why there was some hands up? Just wanted to give it as an example showing why these molecules matter. Okay, and so if we're talking about what's going on in a cell, if you're not able to transfer those fatty acid chains back onto CoA, then you're not going to be able to make any energy from our fat molecules. Okay, so in terms of a CPT2 deficiency, understanding what these enzymes do is really important, right? If there's any problems with that carnitine shuttle, then you're not going to get those fatty acid chains into the mitochondria, so you're not going to be able to generate energy in your muscle cells. Okay, we might actually see hypoglycemia arising because our sugars are going to be the only thing that we can generate energy from. Okay, hypoxia is low energy, low oxygen conditions, so it's not that. And insulin insensitivity, we're actually going to be relying on insulin to help with glucose transport. Okay, so in summary, just for fat breakdown before our break, we have our hormones that are going to be important for the catabolic breakdown of our triacylglyceride molecules. We've got albumin transporting those fatty acid chains. We first need to activate them using 2 ATPs by acyl-CoA synthetase. So synthetase, the name tells you that we're synthesizing our acyl-CoA molecule. Our carnitine shuttle is going to control the entry point for our fatty acids so that they can be broken down in ATP in order to make a huge amount of energy. All right, we'll take a break until 10 after. I'll put this slide back up just in case anybody has any questions. And then we'll talk a little bit more about fat metabolism going forward. That would be confusing. Stu, if you're back there, I need help. Come on. Sorry, I've got technical issues. Give me a sec. Why are you not connecting? I can't get back on.
Sometimes it just takes a second when it's been a bunch of stuff has been plugged in and out of it. Yeah.
Sorry, everybody. When I mentioned that you need a calculator, you definitely need a calculator because even I didn't do the right calculation. So I think I had a 87.5, but it should be 77.5. So 31 times 2.5 gives you 77.5. 77.5 plus 22.5 is how you get the 100. So the final answer was right, but my calc error midway was wrong. Okay, so if it was a short answer question, definitely show your work. Don't jump to the final answer because if you make a mistake like me, we can at least give you part marks to figure out where your calculation went wrong. All right, remember if you don't start with the CoA attached, we subtract off 2 ATPs and a water molecule. All right, so one thing that I do kind of want to talk a little bit about is this asterisk on my summary slide. Okay, so down here at the bottom it says beta oxidation can produce a huge amount of ATP. So we just saw that 108 ATPs were made from a 16-0 CoA molecule. If you had a triacylglyceride molecule, you would have 3 of those, so you would have 3 times the amount of ATP being made. So you can get a huge amount of energy out of these fatty acid chains in beta oxidation. Okay. But the problem, though, is that it's actually dependent on the citric acid cycle, right? We saw that those acetyl-CoAs need to be processed by the citric acid cycle and oxidative phosphorylation. So one of the things that I just wanted to show, I don't know if this is going to work. We've been having sound issues, but I have a little clip from a movie called Run, Fat Boy, Run, which was back in 2007. I know it's a really old movie, but it's quite cute if you haven't seen it. Simon Pegg's one of my favorite actors. And it actually illustrates him hitting the wall. So Dr. DeMello talked a little bit about carb loading, right? If you are running marathons and consuming lots of carbohydrates in terms of building up those carbohydrate stores. Okay, let me see if this is actually going to work. I don't know if the volume, I should ask Stu when he was down here. I'm not sure if this is going to work volume-wise, but let me give it this a chance. I don't want my laser pointer. Oh, it is. Okay.
Do you know Mr. Dawson?
Oh, I'm the assistant coach.
Oh, really? What's your name?
Mr. Coach, that's the hush. Coach. Go. Okay, just call me John. It was me who discovered his potential.
I can't breathe. My head's gonna explode.
Hey, so
I have to stop. No, I'm sorry. You can't. What are you doing? You can't stop. You're nearly there. How much further? It's a little tiny 9 miles. Okay.
Sorry, the volume's not great, but lots of miles to go. Giving him some encouragement to keep going through this marathon.
Oh, no. What? He's hit it.
Okay, baby, let's get you to bed. Jakey. Dennis.
Dennis?
Dennis Doyle, a clothing store security guard running for the last 10 3/4 hours on basically one leg, is refusing to rest until this race is run. Unbelievable.
You've never finished anything in your entire life, Dennis.
All you're gonna want to do is give up.
I'm not the quitter.
That's exactly what you're gonna do, Dennis.
Oh, run away! Run away! Let's go, do.
Dennis?
Oh, I don't believe this.
Yes! He's still going! Go on, you beautiful man! Apologies there, ladies and gentlemen, but it's really rather hard.
All right, so I want to use this. I want you to use this kind of as motivation, okay? When you are feeling down and you need someone cheering, think about this clip in terms of pushing through and finding that, digging deep and finding that motivation to get through and generate that energy that you need to keep going, okay? So when it comes to this concept of hitting the wall, this is where a blend of fuels is really important, okay? So he's been running this really long marathon. He's run out of all of that glycogen stores. And the reason why glycogen is important is because glucose can actually get used to make oxaloacetate. Okay? So remember when we're talking a bit about generating ATP from our fats, so we saw that our fats generate a huge amount of acetyl-CoA. Those come into the citric acid cycle, and it's going to be dependent on the final product, oxaloacetate. Okay? These two molecules are going to join together in order to form citrate to help with the breakdown of our fuel molecules. Okay, so if we don't have a lot of oxaloacetate, then we can't get all of that energy out of those acetyl-CoA molecules. So what we can do is we can actually make that oxaloacetate from either amino acids or pyruvate, and pyruvate can actually come from carbohydrates. Okay, so there's an enzyme called pyruvate carboxylase, which we'll see in gluconeogenesis next week, that can actually help produce oxaloacetate so that your cycle can run even more efficiently. All right, so we really do need a blend of fuels, both glucose, maybe some amino acids, as well as those fats to generate a huge amount of energy, right? So if, for example, when we're talking a bit about that running a marathon and carb loading, right, these carbohydrates can really help you generate that oxaloacetate, right? It can be made from amino acids and proteins too, right? But your amino acids and your proteins you need for your enzymes and from your muscles, right? So we really don't want to be breaking down those. Maybe you have, if there's some excess amino acids, we could use them. But that's where that carb loading is going to be really important to help supplement this cycle, right? And then the cycle is going to run even faster. So let's just say like 100 times faster versus if you only had a single oxaloacetate, then it can't process all that acetyl-CoA that's coming in. Okay, so that's really where this idea of a blend of fuels is important. And when he's hitting the wall, that's when he's actually running out of that glucose from his glycogen stores, and then gluconeogenesis is going to kick in and help supplement and generate a little bit of energy. Okay, so just think about what's going on in terms of the different pathways and how everything comes together. All right, so we're going to switch a little bit, and we're going to talk a little bit about fat synthesis now. Okay, and so you might hear a bunch of different types of diets that might be out there, things like low-carb diets. So I'm just curious what you kind of know about low-carb diets. Can you make any sort of connections to what we're seeing in our course? And if I can get the Mentimeter up, we'll just see what people are thinking about. Okay, so what do you think? How do you think a low-carb diet is going to work? What's sort of the biochemical basis? And what do you think maybe some of these potential side effects might be? How do I show? Oops, that's not what I want. All right, so we've got some ideas coming in terms of things like hypoglycemia. So if you're not consuming a lot of carbohydrates, then you might be in a hypoglycemic state. You're not going to have that many carbs to burn for storage, right? And so it's good for weight loss because instead we can start to burn some of those fats. Low glycogen storage, again, low energy as well, right? Because if you don't have a lot of those carbohydrates, as we just saw with the hitting the wall example, that's not going to help generate efficiently energy from our beta oxidation. Okay? And then things like keto diets, we're going to talk a little bit about ketogenesis next week. There are some side effects that might happen, including that low energy and hitting the wall that we'll talk a bit about. And I think there's another side effects such as keto breath. Okay, so definitely start to pull everything together in order to understand how these things are all coming together. All right, and I'm trying to get my Control P. Why did I lose? Hang on one sec. Doc does not like my computer today. Sorry, Stu. What's the trick to get it back up? Sorry, guys. Display. Come on. Dr. DeMello, are you out there? Stu, if you're out there, can you help me again? It's not mirroring. Every time I switch, it's yeah, I can try. It's not it's this port's not every time I switch over. I'm losing it up here. Oh yeah. All right.
Sorry, didn't help much.
No, it's okay. All right, we're back. Sorry. Okay, so we're flipping over to our anabolic state. Sorry, I can't annotate on the doc. But we're going from our small precursors, so starting, say, with glucose. So we were talking a little bit about our low-carb diets. So now we're going to walk through the pathways that Dr. DeMello started with, talking a bit about breaking down molecules. So we've broken down our glucose. We've produced our acetyl-CoA. We could also be breaking down our fatty acid chains through beta oxidation to make acetyl-CoA. And now we're going to start with this intermediate acetyl-CoA that can be used in order to synthesize our fats, right? And so the whole basis of low-carb diets is if you don't have a lot of glucose, then you're not going to be synthesizing those fats for storage, right? But obviously it depends on how many calories you're taking and your basic basal metabolic requirements, right? And if you have too many calories in terms of fats or amino acids, those all get produced, broken down to acetyl-CoA, and you can still build up fats, right? So it really depends on this idea of a diet and having, more calories than your body is actually requiring. But we're going to talk a little bit about how we go from acetyl-CoA into producing those fatty acid chains for later. So if we want to store those carbons and produce energy from them in beta oxidation, which we saw already. All right, so in terms of our fat metabolism, we already saw that in our mitochondria, that's where beta oxidation is going to take place. But our fat synthesis, that's all going to take place in our cytoplasm. Okay, so in terms of what's going on, really thinking about where these things are happening in a cell. All right, so when it comes to determining sort of what the hormonal response and fat synthesis is going to occur, so I wanted to do a little bit of review. I can't annotate my slides right now, but let's think about this. Okay, so what's the one hormone that we're going to need for the cellular uptake of fuel out of our hormones? What's the main one what's our anabolic hormone? Shout it out. Insulin, good. So write down insulin in that one spot. All right, so insulin's going to help with the uptake of glucose. We saw how it recruits our glucose transporters. It's also going to help recruit and uptake amino acids for protein synthesis, as well as our fatty acids. Okay. And then that's going to start initiating some of our anabolic pathways. Okay, so for the purpose of this lecture, we're looking at fat synthesis. That's going to be an anabolic pathway. So that's one of the ones that's going to be turned on. What other thing did we see was being synthesized in an anabolic process? What pathway or what thing did Dr. DeMello talk a bit about in terms of an anabolic pathway? Glycogen synthesis. Okay, so we've got two pathways that are going to get turned on. So we've got glycogen synthesis, we're going to see fat synthesis. These are all going to get turned on by that anabolic hormone, insulin, and we'll see where that happens in terms of the regulation of our enzymes. All right, so if our anabolic pathways are on, we need to turn off our catabolic pathways. So we're going to turn off beta oxidation that we just talked a bit about. And if we are synthesizing glycogen, we need to turn off, what's the opposite of glycogen synthesis? Glycogenolysis or glycogen lysis, depending on how you pronounce it, or glycogen lysis, glycogenolysis, if you jam those two together. So those are our catabolic pathways that are going to be turned off. There's another pathway that needs to get turned off as well. If we're synthesizing glycogen, what other pathway needs to be turned off? Glycolysis, right? And technically that's not it's a catabolic pathway, but it's going to get turned off. Right? It's going to be on for a little bit. We're going to generate a little bit of energy, but that's what's going to help use that glucose in glycogen storage. Okay? So this is good review. Hopefully this is all coming back to you. Keep on top of it. Keep reviewing. All right, so we're regulating these pathways. We're going to need to turn off our catabolic pathways, right? So there's going to be hormone signaling that'll turn it off. There's also going to be some metabolites or molecules that are going to allosterically regulate our pathways. Okay, so glycolysis is off. I should have included the citric acid cycle, right? That's also going to get turned off. Otherwise, because we've already generated lots of energy, we're going to use that energy for our anabolic pathways. Okay, so the molecules that are going to be important for regulation are going to be those high energy molecules, right? We've already got lots of ATP, lots of NADH. Go back and review, where do NADH and where do ATP inhibit our pathways? Okay, and you'll see that there are steps in glycolysis that Dr. DeMello went through, also the pyruvate dehydrogenase complex and the citric acid cycle. All of these are going to get inhibited by ATP and NADH. All right, so we're going to start synthesizing our fats, and there's going to be 3 things that need to occur. The first thing that needs to occur is we need to get that acetyl-CoA out of the mitochondria, because that's where we were making it, by the pyruvate dehydrogenase complex or beta oxidation. We're going to first export our acetyl-CoA into the cytoplasm. Then what we're going to do is we're going to take that acetyl-CoA and we're going to carboxylate it. Okay, so we're going to add a CO2 group on it, and that's going to form something called malonyl-CoA. And malonyl-CoA is then going to be used by an enzyme called fatty acid synthase, and that's what's going to help build up our 16-carbon chain. All right, so if we're looking at the movement of molecules across the inner mitochondrial membrane, so here's our inner mitochondrial membrane. We already talked a bit about how our acyl chains get imported by the carnitine shuttle. So those acyl chains came in. We were breaking them down in beta oxidation to make acetyl-CoA. Right? We also made some NADH and some FADH2, which can get used for energy. So we're going to make a ton of energy just by breaking down our chains. And then we're going to export that acetyl-CoA out of the mitochondria. And that actually requires the first step of the citric acid cycle. And we're going to form citrate, right? So that oxaloacetate that we saw already is going to combine with acetyl-CoA. We're going to remove that CoA molecule, and now citrate is going to be exported, and then that's what's going to get converted back to acetyl-CoA in order to start fat synthesis. Okay, so we have 2 different ways of moving our carbons in and out of the mitochondria. We've got the carnitine shuttle moving fats in for breakdown, and we've got citrate moving carbons out for fat synthesis. Okay, your citrate will then get converted back to pyruvate, and then the pyruvate comes back in that we saw, similar to what we saw from glycolysis. So there's this nice little cycle where we're not going to lose any molecules, but we're going to be able to get our acetyl-CoA out. And again, there's lots of details in here that you don't need to know. Just know that acyl chains come in on carnitine, and the carbons from acetyl-CoA get exported as citrate and then converted back to acetyl-CoA. All right, so that's all going to happen because CoA is a very large molecule and it can't cross that inner mitochondrial membrane. So we've got citrate synthase that's instead going to create citrate for export. Citrate will then get broken down, and we're going to recreate our acetyl-CoA. That's also going to regenerate a pyruvate molecule that can then go back into the mitochondria. The acetyl-CoA is now in the cytoplasm, and this is where synthesis is going to occur. So we're going to use the acetyl-CoA in order to create our longer fatty acid chains. All right, so that was step number 1. And remember, this is going to be an anabolic process, so we are going to require some high-energy molecules and 2 enzymes. Okay, so the first enzyme is called acetyl-CoA carboxylase, or ACC for short. It's not the Air Canada Centre, but it's ACC, acetyl-CoA carboxylase. And then fatty acid synthase is going to create our longer chains. Okay, and so again, as I mentioned already, acetyl-CoA is going to get converted to malonyl-CoA, and we'll take a look at that reaction in a second. And then we're going to also have this step as our regulated step. So acetyl-CoA is going to be the one where you're going to have to understand how it's regulated, because this is going to decide whether or not we're making fats. So if acetyl-CoA is on, then we're going to be synthesizing, being able to make the precursor we need for fatty acid synthesis. Okay? And it's also going to require an ATP, and then fatty acid synthase is going to require an NADPH as our reducing power. Okay, so this is the first time we've seen NADPH, similar to NADH, slightly different structure. It's got a phosphate on it, but it's also one of those coenzymes that we can use, in this case, for reducing power to drive our anabolic pathways. All right, so we've gotten our acetyl-CoA out of the mitochondria, and now what we're going to need to do is we're going to add on a CO2 group. Okay, so we're going to start with our acetyl-CoA, and as I just mentioned, it's an anabolic process, so we're going to require some ATP. And what we're going to do with that ATP is we're going to use the energy in ATP in order to join on a CO2 group. So just shown in pink, that's your CO2 group, and it's coming from a bicarbonate substrate. Okay, so we're going to transfer a CO2 group onto acetyl-CoA, so onto the methyl tail there to form our malonyl-CoA. So we now have a 3-carbon chain, but it's really going to help as one of our precursors in order to create our fatty acid chain. All right, so we've got this CO2 group that's going to help drive fat synthesis by fatty acid synthase. All right, and so as I mentioned, this step is really going to help with regulation. So this idea of reciprocal regulation. Remember, when one pathway is on, so when fat synthesis is on, we want to turn off fat degradation. And how we do that is that malonyl-CoA that just got made is going to come along and it's going to inhibit CAT1. Okay, so remember, CAT1 was that first enzyme in our carnitine shuttle that is helping move our acyl chains into the matrix for beta oxidation. So if we inhibit that, then our fatty acid chains can't get broken down for energy, and fat synthesis can occur. All right, so we have this committed step, step number 2, where acetyl-CoA carboxylase is going to make malonyl-CoA, and then that's going to inhibit CAT1 and shutting down the carnitine shuttle for the importer chains and beta oxidation. Okay. This enzyme, though, ACC, is also going to be regulated as well in order to determine whether or not we're synthesizing fats. All right, so here on the left, this is our active carboxylase, right? So it's the enzyme that's going to take acetyl-CoA and convert it to malonyl-CoA. It can also be phosphorylated. So another example of enzyme phosphorylation. In this case, we're going to phosphorylate it, change its structure, and now it's going to be inactivated. One of the enzymes that adds the phosphate on is called AMP activated protein kinase. Okay, so there's a kinase that's going to transfer the phosphate from ATP onto our ACC enzyme in order to activate it. Okay. AMP activated protein kinase. Why does that make sense? If there's a lot of AMP in a cell, what does that indicate? Low energy. AMP is low energy. We have no energy. If you have no energy, you shouldn't be synthesizing your fats. You should be breaking them down for energy, right? So in the low energy state, we're going to turn off the enzyme that makes fats. Okay, so yes, we're low energy, we're going to sacrifice 1 ATP, but really that's going to help turn off the enzyme that is synthesizing fats. Okay, so this is our reciprocal regulation. Low energy, we don't need to make any fats. Okay, you can hydrolyze off that, wrong way. You can hydrolyze off that phosphate in order to reactivate it as well. There's another enzyme, so in the catabolic state, right, epinephrine and glucagon. Remember the signaling pathway for epinephrine and glucagon binds to our GPCRs. GPCRs activate adenyl cyclase. Adenyl cyclase activates protein kinase A, and then that's going to also inhibit ACC. Okay, so 2 ways of covalently modifying our enzyme in order to change its structure, inactivate it, and turn off fat synthesis. Okay, then when we're in the anabolic state, that's when insulin takes over. We've got downstream signaling pathway for insulin where phosphatases are going to get activated, and then those can now remove the phosphate in order to reactivate ACC. When you're comparing your pathways, now go back to your glycogen synthesis and degradation, right? Glycogen synthesis is also dependent on phosphorylation, or that dephosphorylation of glycogen synthase. So glycogen phosphorylase was turned on by protein kinase A. Glycogen synthase is also turned off by protein kinase A, that pathway, your reciprocal regulation. Okay, so compare and contrast glycogen metabolism to fat metabolism. Okay, that'll help you simplify these pathways and understand the connection between all of the hormone signaling pathways and the metabolism that's taking place in the cell. All right, so if we're looking at our fat metabolism and its regulation, we've got this step where acetyl-CoA carboxylase is going to be our committed step for fat synthesis. Okay. We see that it's inhibited by phosphorylation whenever you have your catabolic hormones on or low levels of energy. In the anabolic state, insulin signaling pathway will activate our phosphatases to dephosphorylate and activate ACC. Citrate is also an allosteric stimulator, so if you have lots of citrate coming out of the mitochondria, then that tells you you've got lots of substrates that you can use to make acetyl-CoA. That can also stimulate ACC. And then our final product, palmitoyl-CoA or 16-0-CoA, can actually inhibit your active form of the enzyme as well. Okay, so a great way of turning on one pathway and turning off the other. We saw that the product, malonyl-CoA, is also going to shut down fat breakdown by inhibiting CAT1 in our carnitine shuttle. Okay, so go back and take a look at all of your competing pathways and compare them. Maybe set up a table to show which enzymes are on and which ones are off in the various states. All right, so now that we have our malonyl-CoA, it's going to get used by an enzyme called fatty acid synthase. Okay, and fatty acid synthase is a very large complex enzyme. So it's just shown over here on the right, or on the left, sorry, left of your screen. Okay, and it's got multiple different active sites. That's just what all these little abbreviations are showing. And there's actually going to be 4 different reactions that are going to occur in order to create this longer chain. Okay, so we saw beta oxidation had 4 reactions. Fatty acid synthase is also going to have 4 reactions. And again, you don't need to know the details, but I do want to kind of explain it to you so that you have a general appreciation of what's going on, right? And so when it comes to the structure of this protein complex, we have lots of different active sites. So it's a very large protein complex. And we have to have a way of moving our intermediates from one active site to another. Okay, so each of these little what are they, little like arrows, are just showing you where your metabolites are going to go when the reaction's occurring. Okay, so remember there's going to be 4 different reactions that are taking place, and we have to have a way of moving your substrates and products between the different sites. So there's actually something called the acyl carrier protein. Okay, so here you can kind of see the protein structure, lots of helices, some loops, and then it also has a phosphatidylcholine cofactor that's covalently attached. Okay, so another chemical group. So this is a vitamin derivative. We've already talked about how vitamins are really important for metabolism. So here's another great example of where vitamins come into play to help create this structure. And this is part of the larger complex. So this is the ACP area, or the acyl carrier protein. Okay? And so the this long arm actually resembles coenzyme A. Okay, so it has a similar structure to coenzyme A. So what we're going to do is we're going to move the carbons off of malonyl-CoA and move it on to the acyl carrier protein. And now this arm is just going to go moving around our substrates from one active site to another to allow for the different reactions to take place. All right, so there's going to be 4 reactions. Okay, and I did want to draw this out, but I don't know if I can. I'm just going to see if I don't think my laptop's reconnected to the system. So I'm not going to be able to draw it out. Okay, but you don't need to know the reaction mechanism anyways. Okay, but what I did want to kind of show you is the reason why we were creating malonyl-CoA in the first place. Okay, so malonyl-CoA, if you recall, actually has this CO2 group on it. Okay, and so what's going to happen is this CO2 group is going to be really important for helping create a longer chain. Okay, so remember we talked about the spontaneity of CO2 and it's being released is a very favorable thing to occur. When you cut off this CO2, that's what's actually going to drive the condensation or the creation of a longer fatty acid chain. Okay, so the first step that's going to occur is we actually have a condensation reaction where malonyl-CoA is going to get used. We're going to start with an original acetyl-CoA, and then that's going to join our carbons together to form a 4-carbon chain. Okay. Then we're going to see 3 more reactions that are going to take place. This is where NADPH is going to come into play, so you're actually going to require 2 NADPHs each round. And then there's also going to be a water molecule that's going to get released. So it's kind of the opposite of beta oxidation, but it's a little bit different. Okay, and then the next round, we're going to start with another malonyl-CoA. So malonyl-CoA is going to be the donor each round, donating 2 carbons when we get that decarboxylation reaction taking place. Okay, so just in words, this is what you do really need to know. Okay, we need to know that the synthesis is really going to start by acetyl-CoA carboxylase. That's going to create that activated malonyl-CoA that then gets used by fatty acid synthase. Fatty acid synthase is going to repeat the cycles. Okay, and again, we're going to have n divided by 2 minus 1 cycles, because the very first cycle you're going to start with an acetyl-CoA. Okay, you're going to start with a single acetyl-CoA. Each round we're going to add on a malonyl-CoA, and that's going to generate our 16-0 chain. Okay, so we've got 1 acetyl-CoA in the first round. That's going to donate our terminal methyl carbons in our chain, and then each round a malonyl-CoA comes in. We get decarboxylation in order to create our long chain. And then every subsequent round is going to use a 6-malonyl-CoA molecule. Okay, we just need lots of energy for these anabolic pathways. ACC itself requires ATPs, so ATPs to make your malonyl-CoAs. And then fatty acid synthase needs NADPHs, 2 each round. So if it's running 7 times, you're going to require 14 of them. Okay, and you can see that the amount of energy we need to make a 16-CoA chain is not as much as we got when we were breaking them down. Okay, so it kind of makes sense. Any of your dietary fats are first going to get broken down to acetyl-CoA. We're going to generate a huge amount of energy, say that was 108 ATPs. And then if you needed to make some of these chains, say for your membranes, we could use a little bit of that energy, like 7 ATPs and 14 NADPHs. Okay? Those chains could also then be extended. We could have longer chains in our membranes. That's all going to happen in the ER. And you're going to use more malonyl-CoA as well as more NADPH. And when we talked about our lipids, remember we talked about them sometimes having these double bonds. Those enzymes that are going to play a role in that are called desaturases. So whenever we're creating our chain by fatty acid synthase, it's going to be in the saturated form. So if you want to add in those double bonds, you're also going to require some energy as well as oxygen. All right, so when you're going back and you're reviewing, you're trying to compare and contrast the different pathways. Okay, so we have our synthetic pathways happening in the cytoplasm. We've got our breakdown pathways happening in the mitochondria, right? Beta oxidation taking place in the mitochondria. That's our catabolic pathway. Beta oxidation was making acetyl-CoAs, whereas fatty acid synthase is going to use one acetyl-CoA as well as a malonyl-CoA each round. We have different coenzymes that are going to be taking place, that are going to be used, right, or produced. So fatty acid synthase needs NADPH, whereas beta oxidation was producing NADH and FADH2s. Okay, so different coenzymes are required there. And beta oxidation has 4 different separate enzymes, whereas fatty acid synthase is this one large complex protein enzyme. Your number of rounds are going to be the same. Oops, I don't have that animated in. But that's going to be the same for synthesis or degradation, right? That n divided by 2 minus 1. Right? If you're trying to figure out how many times are these things happening, it's going to be a similar process whether you're synthesizing fats or breaking them down. All right, the last thing I want to talk a bit about is how we actually get back to that storage form for our adipose tissue or your fat tissue, right? Remember triacylglycerides, that was the structure that we wanted to store our carbons for later. So in order to create that molecule, what we're going to do is we're going to start with phosphatidate. So let's go back and review the structure of our glycerol phospholipids, so our glycerol backbone. Phosphatidate has 2 fatty acid chains already, as well as a free phosphate group. The first thing we need to do is get rid of that phosphate group so that we can stick our 3rd chain on there. So we're going to hydrolyze off that phosphate. That's going to make diacylglycerol, so glycerol with 2 fatty acid chains on there. And then it's going to be an acyl transferase that's going to transfer that 3rd chain onto the 3rd position. Okay, and that's what's going to make our triacylglycerol molecule. When it comes to thinking about regulation and our signaling pathways, whenever you have insulin signaling on, that's also going to help activate all of these enzymes at the level of gene expression, right? So we're going to create more of our acetyl-CoA carboxylases, more of our fatty acid synthases, and then those acyl transferases to stick that 3rd chain on. Okay, and all of this is going to happen in your liver. And that's, we're going to talk a bit about that on Thursday in terms of the production of lipoproteins, which is where we're going to release them in the bloodstream for use. Okay, so apologies for sort of the technical difficulties, but definitely go back, start staying on top of the material and reviewing on a regular basis so that you can compare the hormone signaling that's important for these pathways, as well as the regulation. So you have one pathway on and one